Good evening, everyone from the US, and good morning to those of you that are in Australia. My name is Suzanne Barnett, and I'm hosting your webinar for you today. Now, before I get into the webinar, I just need to know that you can all hear me perfectly fine. So, as usual, if you could click on your hand button, that would be great. Thank you. There's one that's gone up, so just waiting for a second for some of the others. Or if you could type in the question box, if you can't find your hand button. We've only got one hand that's gone up, so I'm just going to wait for a second to make sure. Perhaps, Vivian, thank you. Janie, Becky, great. Andrea, thank you very much. Okay, so then I can assume that others can hear. Okay, perfect. And as usual, just to remind you, if you switch off any of your other applications, it just ensures that you get the better quality broadcast of this webinar. And of course, they are all recorded anyway. So once again, we'll be aiming to finish by 1 p.m. and that will give you an opportunity to type in any questions. So do type those questions in. We will be asking them at the end. So anything that comes to mind, just ask those questions. Okay, so our topic today in the SNP Learning Series is Presentation 6, and we're talking about serotonin. Once again, our speakers are Sterling Hill, Carolyn Ladowski, and Cynthia Smith. And the first part of our presentation today will be presented by Carolyn.
Hi everybody, thanks for joining us. We got quite a good topic today. We were all just saying how it was really nice to prepare for this. So the serotonin pathway is great, and we're going to talk about the dopamine in a couple of weeks. But just to refresh your memory, the dopamine really does play important roles in maintaining alertness, while serotonin is really our relaxation, our appetite control, and certainly our sleep cycle. So we need obviously a healthy presence of both compounds, and we need the cofactors in place. And it's really important to understand that there are actually many steps in each pathway that gives us really good opportunities to provide nutritional support to help it out, because we obviously need our amino acids. And this is one of the issues we get with a lot of patients, is their overall protein content is quite low. And so that is definitely going to have an impact on their neurotransmitter synthesis. We've got to have our vitamins, particularly our B6 and our zinc, when we're talking about any neurotransmitter pathway. And we need phytonutrients that will help stabilize our neurotransmitters. So, as we know, serotonin synthesis begins with L-tryptophan, and that is hydroxylated to 5-hydroxytryptophan, and we know that that needs our 5-methyl, our B6, and our vitamin C, zinc, and magnesium. And once this is released into the synapse, serotonin activity depends on healthy uptake balance. So what we're going to do today is talk about all the SNPs that affect serotonin synthesis, and some of the things that you may see in patients presenting with either high or low. So here's our serotonin and dopamine pathway, and you can see that dopamine and serotonin are synthesized in the presynaptic neuron, which is the blue, and these pathways require our amino acids and cofactors, and when it's released into the synapse, they have to bind to their receptors on this postsynaptic neuron, and that's the green area, and that's how they relay their messages, and it supports obviously all those areas of mental health and our emotional well-being. So let's have a look specifically then at the serotonin pathway. And again, thanks to Ben Lynch for these pathway planners, and you can get copies of these on his website, seekinghealth.org. So have a look there, and you can see at what we call our biopterin pathway. And obviously we need that very important MTHFR gene to be working, and to enable the recycling of biopterin to tetrabiopterin. And I'm going to talk about that in a little while. But then we need that conversion from tryptophan to 5-hydroxytryptophan and then to serotonin. So that's the pathway we're talking about today. And we're also going to be looking at the kynurenine pathway, which is that side chain that goes from tryptophan. And basically what it does is cope with the overload of tryptophan that the rest of the pathway doesn't cope with. And Cynthia is going to talk to you about the sulfation of these pathways, particularly the ones that you can see there down the bottom. This is Cynthia's pathway, and I'm sure she'll take you through this, but it's a great one to really look at all these genes to show you from the DHFR right through. So on the right-hand side, you obviously see your serotonin, and the left-hand side, you can see your dopamine component. And again, you can see those genes and how they play out into contributing to our serotonin synthesis. So Sterling is now going to take us through these 4 key genes in that first part of the pathway, and she'll certainly give you some great insight into this DHFR. So Sterling, I'm going to hand it over to you.
All right. Before I start talking, I'm going to go more on the serotonin part of these genes. I'm not going to explain the genes since we've basically learned about many of these already. So DHFR, one of the alternative pathways for the final 2-step reaction for BH4 biosynthesis is DHFR dihydrofolate reductase. Dystonia dopa responsive due to sepiapterin reductase deficiency results in decreased central production of BH4. So patients with SPR deficiency have central nervous system manifestations, but they do not have hyperphenolaniemia, since peripheral production of BH4 can be compensated by alternative enzymes in the final 2-step reaction. The cerebral neurotransmitter deficiency in patients cannot solely be explained the low normal BH4 production in the brain. The relative absence of DHFR in the brain results in the accumulation of 7,8-dihydrobiopterin, which is a competitive inhibitor of tyrosine hydroxylase and tryptophan hydroxylase, also adding to the reduction of dopamine and serotonin synthesis. So DHFR plays a very important role in serotonin. TPH, the tryptophan hydroxylase gene, makes the TPH enzyme. This enzyme is involved in the production of serotonin. Scientists searching for the genetic roots of depression have often focused on genes related to serotonin, and TPH is among the ones that have been singled out for study. The TPH enzyme transforms a dietary amino acid called tryptophan into 5-HTP. Then 5-HTP is converting in the brain to 5-hydroxytryptamine, the chemical name for serotonin. Variations in this gene may lead to dysfunction in the serotonin production process. DDC, the dopa decarboxylase enzyme, catalyzes the synthesis of dopamine, tryptophan hydroxylase 2, a brain-specific rate-limiting enzyme in serotonin synthesis and brain-derived neurotrophic factor, a growth factor that interacts with serotonin to regulate growth, survival, and plasticity of serotogenic neurons. AANAT, arycholamine N-acetyltransferase, is known to catalyze the acetylation of serotonin, a rate-limiting process in melatonin synthesis. Different singular nucleotide polymorphisms in the AANAT gene were identified recently in the Japanese population, and one of the genes was significantly associated with the delayed sleep phase syndrome. Okay, Carolyn, back to you.
Okay, thanks, Sterling. So let's have a look at those in a little bit more detail. So DHFR, as Cynthia says, as Sterling says, when you look at those pathway planners, if you have homozygous SNPs, not only is it going to affect your folate metabolism, but it's going to affect the whole issue of your biopterin recycling. And we want BH2 to be converted to BH4 to enable those substrates into that phenylalanine, tyrosine, tryptophan, and arginine pathways so that we can get conversion of our neurotransmitters. And I'm going to talk quite a bit about the issues around tetrabiopterin recycling, because it is a key issue for us to get our heads around. And quite frankly, we could really talk about this for a whole webinar. But if we look at tetrahydrobiopterin is not an essential nutrient because it can actually be synthesized in human tissue, and that's predominantly from guanosine triphosphate, or we see there GTP. The key roles of BH4, it's a cofactor for the hydroxylation of our aromatic amino acids, so phenylalanine to tyrosine, tyrosine to dopa, and tryptophan to serotonin. And importantly, it's a cofactor in nitric oxide production from arginine. So you can see that under normal conditions, this BH4 availability is really maintained by our GTP gene and DHFR because it has to be recycled. And you can see there that DHFR gene converts BH2 to BH4. So on the right-hand side, you can see that uncoupled NOS is characterized by the production of superoxide. And so when we refer to NOS uncoupling, it's because it is promoted by a decreased BH4 availability relative to our BH2. And when we get this, we're producing more superoxide. And that reacts with nitric oxide and forms our peroxynitrite. And it's peroxynitrite that is that really reactive anion that oxidizes BH4. So in your pathway planner, you can actually see that peroxynitrite inhibits BH4. And so that's a really big issue for us, because in oxidative stress, BH4 is reduced to BH2 and we get this more uncoupling. And DHFR can reduce these BH2, and so what we should be doing is getting regeneration of BH4, but we're not. So I guess the critical take-home message for all of you with this whole BH4 story is that prolonged oxidative stress causes superoxide and peroxynitrite elevation, and that causes uncoupling of NOS, which means we get a shift of NOS activity more towards superoxide and less towards nitric oxide production. And so that's what we mean by NOS uncoupling. And this increase in peroxynitrite shift means that we're just superoxide is taking over. And we all know that nitric oxide is crucial for our epithelial. We remember we talked about that in relation to our hydrogen sulfide as a key gas emitter. So we need it to ensure that we have our great epithelial contraction, vasodilation, control of blood pressure. And, we also think that if we've got peroxynitrite elevation and oxidative stress, we're also going to have NF-kappa B activation and more inflammatory markers. So if you look here, so as we said, BH4 is an essential cofactor for the production of these neurotransmitters, including serotonin, and you can see that pathway there. So let's talk a little bit about peroxynitrite, because it's a key thing, and as we said, it inhibits our BH4. So this whole pathway is really interesting because we know infection really stimulates the synthesis and indeed its secretions of inflammatory cytokines. And several of these induce an enzyme that we call inducible nitric oxide synthase, or INOS. You'll see that written as. and it leads to an increase in nitric oxide and its oxidant product, peroxynitrite. So if we then have a look here, you can see that nitric oxide reacts with superoxide to form peroxynitrite, and this causes elevation in NMDA activity, and it increases nitric oxide and peroxynitrite. So if we've got excess peroxynitrite, we know that that's a really potent oxidant, and we're going to be increasing our oxidative stress, and this will lead to NF-kappa B stimulation, which will increase production of our 5 inflammatory cytokines, particularly interleukin-1b, interleukin-6, interleukin-8, tumor necrosis factor alpha, and also our interferon, our tumor necrosis factor gamma, and INOS. So that's pretty key, isn't it? Because all these chemical messengers, we know they're involved in inflammation of tissues, and even NF-kappa B, can be increased by bacterial and viral infections and oxidative stress. And so this is going to lead to things like oxidation of our lipids and proteins. It can lead to DNA damage, cellular energy depletion, cell death even, because it causes a mitochondrial calcium efflux, and that will cause cell death. So if we summarize, peroxynitrite will also inhibit our mitochondrial superoxide dismutase. So it's important when you're thinking about people you see people with the SOD, or what we call SOD mutations, they're going to be more susceptible to this because SOD is not as effective. So we need to support people with the SOD genes with things like manganese and zinc, because if we suppress our superoxide dismutase, we're going to get more peroxynitrite formation, and we don't want that. And it's this peroxynitrite that really is involved in our NMDA receptor neuronal cell death. So that's important because so many of our patients present with what we believe this elevation in peroxynitrite, which is really affecting their neurological function. And so that brings us on to the NMDA receptors. They're found in the brain, the spinal cord, the peripheral nervous system. And don't forget, they're a class of glutamate receptors. So when activated, they're going to cause excitatory neurotransmission. And because they play a role in memory function, they have been linked to things like Alzheimer's. And they're going to be stimulated by anything with glutamate, aspartate, and even glycine. So, things that we should be talking to our patients about is minimizing foods that have that glutamate excitatory behavior. So even things where people think they're doing the right thing and they're having lots of bone broths and things like that, it's not necessarily the right thing to do because they're going to increase their glutamate levels. Glutamate is not just in MSG, but it's in a lot of different products, even gelatin. So many people are having, great gelatin thinking they're doing the right thing, but it's actually stimulating these NMDA receptors. So this represents a typical NMDA receptor, and these contain basically 4 subunits, and you can see 2 glycine binding NR1 subunits and 2 glutamate binding NR2 subunits. And these really allow for this, we were talking about, this cationic influx from the synaptic cleft into the cell. And we know that these NMDA receptors really play a key role in this signal transduction in certain areas of the brain. So we need these not to be working really well, so that we're not getting a neurological inflammation. And copper really acts as a modulator of this neuronal transmission in our central nervous system. So we've got to balance, and that's one of the reasons why, particularly when we see anxiety, when we get huge disturbances in copper and zinc, if you've got really elevated copper, it's going to be affecting these NMDA receptors. We've got, excessive NMDA receptor leads to xanthine oxidase activity that will generate superoxide. And excessive NMDA stimulation, as we said, is related to anxiety, depression, Parkinson's disease, Alzheimer's disease, ALS, and AIDS-related dementia. And in some studies, even SLE and Huntington's. So we've seen this before, but I thought we just put it up again. Some of the issues around DHFR, nothing relating, there's not a lot of research that, puts it where we're relating it directly to serotonin, but I think that's just a matter of time before we see that. So let's have a look at IDO. Now, IDO isn't in your on your variant report, but it will be. So we thought it's probably good to just have a quick look at this particular gene enzyme group. And basically the immune system, the serotonergic system, and the kynurenine acid pathway are all linked by IDO. So the theory is that upregulation of the immune system, particularly when we've got interleukin-2 and interferon gamma, then we are creating we're creating inflammatory cytokines, and our Th1 activates IDO. And if we look at IDO, it's directly involved in the breakdown of tryptophan. And we know that that is the precursor to 5-hydroxytryptophan. And so the activity of IDO is going to be enhanced by inflammatory cytokines. So we get activation of IDO via stimulation of our T cells, and it will deplete all available tryptophan. So we often see, and there's a lot of research about this now, which is great, that often people who are really sick, they get depressed, and this is the mechanism behind it. So our cytokines really influence our tryptophan metabolism within quite a short amount of time, and will then affect the downstream pathways, and obviously quinolinic acid is one of those. Now, I'm sorry that this is a bit hard to see, but I guess the key thing I just wanted to show you is the catabolism of tryptophan to N-formyl kynurenine, and it's this enzyme that acts on multiple substrates. Our D-tryptophan, our L-tryptophan, 5-HTP, tryptamine, and serotonin. And this pathway will lead to either quinolinate or nicotinate, depending on the tissue location. So in the liver, it will lead to production of nicotinic acid, but depending on timing of signals, this pathway seems to either activate or inhibit neuronal responses to acute and chronic stress. So this pathway is B6 dependent, and it's important to remember that the quinolinic acid really provides a very important link between the immune system and the brain, because increasing quinolinic acid really stimulates our glutamate excitatory behaviour. So our IDO pathway, this is really just to show how it decreases the local concentration of free tryptophan. That's what it's meant to do. And if we've got acute stress, then it's going to affect our T helper cell. And this is a good one because it shows you that the liver really regulates tryptophan levels in plasma, and it's through this kynurenine pathway. And it's initiated by the heme enzyme IDO. So if you've got postprandial tryptophan in excess and it can't be utilised for protein synthesis, then it's going to enter this pathway and it will be converted to nicotinate. That's what it should happen. However, this kynurenase conversion to our 3-hydroxykynurenine, and you can see that on the left-hand side, it's B6 dependent. And so if we've got a decrease in B6, it means we're going to get an accumulation of 3-hydroxykynurenine, and that will lead to an elevation of quinolinic acid. So that's key. So this whole pathway basically looks after our excess tryptophan, but if we're low in B6, you're going to be shunting more into quinolinic acid rather than nicotinate. So that's really key. So if we look at quinolinic acid, it is a really critical link between the immune system and the brain. It interacts with our NMDA receptors, and it may cause glutamate excitatory behaviour. They have actually even linked this with Alzheimer's disease. And exposure to phthalates will actually also increase quinolinate production. The end benefit is NAD, and I've started to use NAD more and more and more, or just good old-fashioned B3, and you know, it has such a huge bearing on some people's mental capacity, just to calm them down. And I really do think it's because this whole kynurenine pathway is disturbed by gut bugs, gut infection, and we see this a lot. And it can be indicated both IBD and neuronal degeneration if we're seeing elevated quinolinic acid on organic acid reports. So the TPH pathway, really just to show you that the L-tryptophan relies on two key enzymes, the tryptophan hydroxylase, which is the TPH, and the amino acid decarboxylase, the DDC. And as Sterling said, these two are key in the conversion to serotonin. And serotonin, you know, orally, when we eat it out of food, doesn't really pass into the serotonergic pathway of the central nervous system because it really can't cross the blood-brain barrier. However, tryptophan and particularly 5-HTP can and do cross the blood-brain barrier. DDC, really just to show you that it has an effect on MAO as well as dopamine. We're going to talk about this a lot more when we discuss the dopamine pathway next week. And again, same things. We will go into these in detail because it really does have more of an effect. From a research perspective, we're seeing a lot more of it related to dopamine dysfunction. AANAT, key regulator of our day-night cycle, so our circadian rhythms. And you will see on the pathway planner, it's the key gene that converts serotonin to melatonin. So if you are seeing homozygous SNPs in this particular gene, then melatonin deficiency is probably a key factor. And you know, as we all know, you can try melatonin with some people and it works beautifully, and others it has absolutely no effect. But I'm more and more convinced that really people with this AANAT really do need some help with melatonin synthesis. And if you can at the same time increase serotonin production, that's obviously going to have a good effect. It also catalyzes the N-acetylation of serotonin, and that's obviously key in our melatonin production in the pineal gland. And just a bit, you know, there is one study here that does link this with depression. So I thought that was pretty interesting. All right, so I'm going to pass you on to Sterling now, who is going to take you through these second lot of genes in the serotonin pathway, and most importantly, the MAO. So Sterling, over to you.
Hello. MAOA. The MAOA gene is located on the X chromosome and encodes the MAOA enzyme, which metabolizes the neurotransmitters such as norepinephrine, serotonin, and dopamine, rendering them inactive. In one study I pulled up, maltreated children with the genotype conferring high MAOA activity were significantly less likely to develop antisocial behaviors problems, and then maltreated children with low MAOA activity genotypes, total opposite. Now, I know we've seen a lot of people, children with autism, in their neurotransmitter testing usually have, most of them have very high serotonin. And then we've seen people with depression, again, the low serotonin. Fixing the gut is very important for serotonin. And some different things that we know how to get serotonin so we can go to bed at night, sleep, make that melatonin. Walk in the grass, barefoot if you can. Earthing and grounding is really important for your circadian rhythm. Also, blue light glasses at night. If you're someone like me that has to research once the sun goes down, I like to put on blue light glasses for about 20 to 30 minutes, and they're very cheap, very, very cheap to get. And that helps your brain to tell you that, hey, all lights are off, and your body will start breaking down what serotonin you have into melatonin. Also, no electricity in your bedroom if at all possible. That goes for alarm clocks too. Putting your cell phone on airplane mode, turning off your Wi-Fi at night helps a lot. Also, ditching baby monitors. They're very, very unhealthy for babies. Very unhealthy. I actually got to talk to Jack Cruz one time. He's spoken with TED. He is very interesting. He studies quantum physics. He's an oral surgeon and a neurosurgeon here in New Orleans. And he actually had a baby once that was up all night crying, and he told them one simple thing: get rid of the baby monitor. The baby slept its first full night in months. Now, also, a little keynote about grounding yourself is really important with serotonin and melatonin. People in airplanes are never grounded, and one of the ways you can, and it can disturb your sleep pattern, so sometimes it's not only always the time shift of you going over a continent or time zones. Sometimes it has a lot to do with you not being grounded for quite a bit. And another thing I learned from Jack Cruz, what you can do is put on stockings, like some stocking socks, and instead of putting your foot on the plane, on the footstool rest, the plastic port, the two metal boards that connect the plastic footrest, put your foot there. Also, you can wear leather-soled boots or leather moccasins, leather-soled moccasins. This helps out a lot, especially in the winter when you can't walk barefoot outside. That also helps with grounding. Very important little keys to help break down serotonin into melatonin, since we're talking about MAOA. And something I like to do in the winter, knowing that I don't get too much sunlight, is I make a pot of chicken soup every week because I know it contains tryptophan. And then I also have organic pumpkin seed butter. It's loaded down with tryptophan, very high in tryptophan. In turkey, here in America, it's kind of hard to find turkey without carrageenan in it, which is a carcinogen. But if you can find it, it's a great source of tryptophan. All right, ALDH2. That is the aldehyde dehydrogenase 2 and serotonin 2A receptor gene. And they are involved in metabolizing serotonin and in encoding serotonin receptors. ALDH2 interacts in bipolar 1 disorder, but not bipolar 2 disorder. Okay, let's see. ADH1B. ADH1B is class 1 alcohol dehydrogenase 1 beta subunit. This gene, along with serotonin, holds an association with whether or not a person may become alcohol dependent. Serotonin is a neurotransmitter that is important in mood regulation, and it is associated with decreased alcohol consumption. Now, the SLUT genes, 1A1, 1A2, 1A3 pathway is involved in serotonin degradation. Savannah-transferase has been identified in both the central and enteric nervous systems. And the isoforms SLUT 1A1, 1A2, 1A3, and 1C2 have been shown to sulfate serotonin. In addition, 7-hydroxylated serotonin may be formed as a result of cellular oxidative stress and subsequently sulfated. Back to you, Cynthia.
Okay. So the next slide is Ben's pathway planner again, and I just wanted to point something out. There's been some corrections to this drawing. If you look where at the top under FLOR2 to DHFR, and I did the same thing initially, but I've updated my drawing in the compendium. Phenylalanine doesn't flow to tyrosine, doesn't flow to tryptophan, doesn't flow to arginine. Those are all discrete amino acids. But what is shared in common is that BH4 is needed for the TH enzyme, and BH4 is needed for TPH enzyme. So it's hard to draw it, and if you want to make it look cool, but the reality is those things don't make each other. So that's one thing. The other thing is that we talked about, Sterling talked about ALDH 1B1, and you can see that below if you follow the pathway from 5-HTP to DDC to serotonin. ADH1B, or that should be AL, I think. Anyway, these enzymes, ALDH1, ALDH2, ALDH1B1, and 3A2, are also involved in acetaldehyde breakdown, which is the second step of breaking down ethanol. And so what you see here is an interplay. Some of the same enzymes that help us break down alcohol also help us break down serotonin. Now the next slide is the same slide that Carolyn talked about earlier. In this slide, again, you can see that some of these enzymes that share alcohol breakdown also share serotonin breakdown. One of the things you also have to know, and it's not really shown on any of these slides, is that acetaldehyde, as acetaldehyde goes up, and these ALDH enzymes, if there are SNPs in them, what will happen is you'll start stimulating DRD2, which is a dopamine receptor. We'll talk about this next week, but you're also inhibiting your methylation. So there's a feedback to inhibit methylation. The other thing is the cofactor for these ALDHs is NAD. So that's the vitamin B3 derivative. So those things are really important. A lot of these pathways for the neurotransmitters require output from the Krebs cycle. So you have to have a functioning Krebs cycle, which means you have to have sufficient vitamin B1, B2, B3. That's just for the Krebs cycle. And then, of course, you need B5 and B6, and iron is another cofactor. Now, the next slide is similar to one that Carolyn talked about. Serotonin has to be broken down. And MAOA is one of the primary breakdown enzymes, in addition to the ALDHs. MAOB is also involved in the degradation of serotonin, but it's typically associated with dopamine. But MAOA and MAOB break down serotonin and dopamine. It's just MAOA is more associated with serotonin breakdown, and MAOB is more associated with dopamine breakdown. Now, there's a lot of research into MAOA. And remember, this is a SNP here comes from the mother. There's a lot of studies about autism, ADHD, anger control. In fact, when I first started studying all of this, MAOA was considered the warrior. They called it the warrior gene. Increased risk of suicide, increased risk of gout, and things like that. But when you look at the research, you're not just getting one SNP and one enzyme increasing the risk of something. There's always other things that interplay. And so you have to look at receptors, you have to look at carrier proteins, you have to look at a lot of things. Here are just some more MAOA SNPs. And you can see all of this has to do with dysregulation of serotonin and also dysregulation of dopamine. So you'll see in the second one here, increased risk of Parkinson's disease. And Parkinson's disease is associated with the dopamine pathway. Here's another slide associated with lipid peroxidation and ALDH, alcohol consumption. So again, alcohol breakdown is a two-step process, and the first step can either be catalase, the use of catalase, or it could be the use of ADH, which is an enzyme that's active in light drinkers, or it's one of the CYP2E1, which is active in heavy drinkers. So that's step one, but they end up in acetaldehyde. And then after that, they need to be broken down. And like I said, they share some of the same enzymes that are needed to break down serotonin. So people who are drinking heavily could have issues with their neurotransmitter status, specifically serotonin, and again, also their sleep. I think this looks like this is a mitochondria with an inner and outer membrane. Diseases in which activators of ALDH may be beneficial, and so you see all of these different diseases associated with ALDH2 SNPs. It's far-reaching, so again, alcohol drinking affects all of these. Here's some more research about the ALD2 SNP functions. First one, of course, is alcohol metabolism, and then there's a link between gastric cancer and alcohol. Colorectal cancer, esophageal, gastric, we all know about those with increased drinking. Head and neck cancer. And then the RS441 is associated more with the breakdown of dopamine, as is the next one. So you see how these cross over between both the serotonin and dopamine pathway. SULT, we've talked about these. SULT enzymes catalyze sulfate conjugation in phase 2 liver detox. Diseases are endometrial cancer and colorectal cancer, and both of those are associated with estrogen because these sulfation enzymes in phase 2 liver detox are very active in breaking down estrogen. So we have the three pathways. We've got our methylation pathway that's active in breaking down estrogen. We've got SULT, and then we've got, of course, glucuronidation. So those three pathways are associated with hormones. And so SNPs and the SULTs will often be associated with issues that have to do with poor breakdown of hormones. This is another slide. We've seen this. We saw this last week, showing that the SULT pathway is active in many organs, but primarily in the liver. The next slide talks about tamoxifen. Now, tamoxifen, big pharma, is a non-steroidal anti-estrogen, and it's one of the most commonly used drugs to treat breast cancer. Estrogen is involved in the growth and development of breast cancers, but I want to clarify something. It's not the estrogen itself. It's the poor breakdown of the estrogen, where the estrogen intermediary metabolites recirculate and add on top of your own estrogen. And so the estrogen alpha receptors have to deal not only with your estrogen, but also this recycled estrogen intermediary metabolite. And you often see that with that CYP1B1 and the COMT downregulation. One of the things about this slide that you'll notice too is that tamoxifen itself as a drug has to be broken down by its own set of enzymes. CYP3A4 is one, but also CYP1A2 is another one. So people with downregulations in CYP1A2 often have a harder time with tamoxifen because it stays at a higher level in their circulation because they're not breaking it down well. Here's another slide about SULT1A1 SNPs and the risk allele. Again, SULT is also related to bile recycling, so keep that in mind as well if somebody's having gallbladder issues. SULT1A3 belongs to high affinity glutamate transporter family. There's research here associated with ataxia, peripheral artery disease, and shock syndrome. But one of the things I want to say about these SULT SNPs, again, alcohol plays a role in this, as does ALDH, so keep that in mind. Since this is mentioning glutamate, I wanted to say something else. Glutamate receptor stimulation, overstimulation, we have to have glutamate receptor stimulation to stay alive. Overstimulation causes all kinds of neurotoxic effects. And I just finished a series, and I'm sure a lot of you heard it. It's a alternative cancer series. And one of the things they brought up is that glutamine that we put in our smoothies, which I did for years to help heal our gut, can convert to glutamate. And we have glutamate receptors not only in our brain but all around our body. And what the research is showing is that besides glucose, glutamate is also very stimulatory to cancer cells. Here's a nice little picture about SULT1A3. And this will lead us into Carolyn's talk in just a moment. This is one of the pathways that breaks down acetaminophen. Another pathway that breaks down acetaminophen is glutathione conjugation. So overdoses associated with acetaminophen are sometimes associated with liver failure. So what we're finding here in the U.S. is that giving acetaminophen to children after they've had vaccines without knowing their genetics is not a good thing, because acetaminophen is very difficult for some people to break down based on their genetics, and SULT is one of them. And again, another one is glutathione conjugation and also glucuronidation. So that would be the UGT enzymes. Now, Carolyn mentioned NOS inducible, NOS1, catalyzes the synthesis of NO. It requires tetrahydrobiopterin, and she spoke at length about BH4 or tetrahydrobiopterin. Anytime there's a stress on nitric oxide synthase, whether it's from SNPs, and mostly NOS3 though, whether it's from SNPs or missing cofactors or low arginine, or this need to be breaking down ammonia. We talked about the CBS pathway a couple classes ago. Breaking down ammonia puts a stress on nitric oxide synthase, which in turn, besides requiring arginine, also requires BH4. So another way you can deplete your BH4 is by having to break down high levels of ammonia. Whoops, NOS1 is not the inducible. NOS1 is the fetal. So NOS2, this is the inducible. And if you look at the slide, it talks about lipopolysaccharides and cytokines and inflammatory diseases. Now, lipopolysaccharides are one of the things they test for when they're looking at leaky gut potential, because lipopolysaccharides are associated with some of the bad gut bacteria, and those lipopolysaccharides can get into our immune system and upregulate it if somebody has a leaky gut. So going back to the IDO enzyme, inflammation induces the IDO enzyme, which kind of cannibalizes the tryptophan and takes it away from making serotonin. And Carolyn talked about the connection between NOS2. So these things all interact. And one of the drawings that I like to use when I'm talking with clients is Dr. Yasko's drawing, because it shows a big picture and not a little pieces like this. So inducible NOS, or NOS2, is a reactive free radical that acts as a biological mediator. So we don't want to induce NOS2. NOS3 is the one that everybody knows about. This is nitric oxide synthase 3. This is the one that synthesizes nitric oxide and citrulline, which is what we want. We want nitric oxide and citrulline. That means we have enough BH4, we have enough arginine, and we're making nitric oxide and citrulline. When we don't have enough BH4, for example, because we're not methylating well, then you start to see these peroxynitrates. And that, in turn, puts stress on the superoxide dismutase genes. So you'll see here that the cofactor is also FAD, so we have to have a good, working good mitochondria function, electron transport chain, and Krebs cycle. This one is implicated in dilation of our blood vessels. Mutations are associated with Alzheimer's, hypertension, stroke, preeclampsia. So the Alzheimer's connection, yeah, that's there, but it's Alzheimer's is actually more than that. It could be not only the fact that the blood vessels are not, dilating and contracting properly. There's a lot more factors associated with the Alzheimer's, but the stroke and the hypertension and the preeclampsia. There's something that we haven't talked about. Nitric oxide, of course, produces nitric oxide and citrulline, but the nitric oxide, again, is implicated in vascular smooth muscle regulation through a cGMP-mediated signal transduction pathway. Nitric oxide, in turn, mediates something called vascular endothelial growth factor, or VEGF. And that's associated with angiogenesis in coronary vessels and promotes blood clotting. People who have attended any of these anti-aging seminars would have heard quite a bit about VEGF, so it's a lot of hot research there. Here's another drawing of the NO pathway. And it's implicated, again, in high blood pressure, right, because it's vasodilation, aneurysms, pulmonary hypertension, strokes, myocardial infarction. So again, we're talking about the dilation and relaxing of the endothelial, the blood vessels. But I, again, would encourage you to take a look at Dr. Yasko's drawing, where it's very clear to see that link between high ammonia, nitric oxide, BH4, arginine, and then peroxynitrates. So nitric oxide and its metabolites, nitrate, which is NO2, and nitrate, NO3, are potential biomarkers. The basics of nitric oxide metabolism are highlighted in pink, including the formation via nitric oxide synthase and the reduction of NOx. Pathological processes that are known to modulate NO metabolism are shown in the green box. And then the uses for NOx biomarkers are shown in the light blue box. Here are some risk alleles associated with NOS1. So, for example, carriers of the first risk allele C had a 1.7 5-fold increased risk of Hodgkin lymphoma and a 3.0-fold increased risk of follicular lymphoma if they were low vegetable and fruit intake, but a 60% reduced risk in follicular lymphoma if they ate their fruits and veggies. And again, there's more research here associated with Hodgkin's lymphoma, of course, heart disease, Parkinson's. There's one, then on the next slide, there's one risk allele associated with NOS2. There were two on the previous slide. And then NOS3. And you can see a lot of the things that have to do with NOS3 are the heart disease and blood vessel function. Now we're going to talk about PAPs next, and this is a very interesting area. Carolyn's going to talk to you about PAPs and how it relates back to these pathways.
Okay, thanks, Cynthia. So after putting together these few slides on PAPs, I realised that it's probably a whole another presentation in its own right because it's just so interesting. Okay, so if we have a look, I'm just going to mute Cynthia. Okay, if we have a look, we've been hearing that sulfation is so crucially important in inactivating certain things, particularly xenobiotics, neurosteroids, hormones, and our catecholamines. Remember that PAPs, which is 3-phosphoadenosine 5-phosphosulfate, is our universal sulfate donor, and its synthesis is really the limiting factor in sulfation. So that's why it's so crucially important. We need inorganic sulfate, and we need 2 AT molecules for this to function. And predominantly the 2 enzymes that make it work are ATP sulfurase and ATP kinase. Now, it's decreased if we have low sulfur diet. So remember when we were talking about CBS pathway, we were saying that really a low sulfur diet is not a great idea. And you'll see why, because this has far-reaching implications, not just for serotonin, but for so many other things in our diet. And xenobiotics also will decrease. So, and predominantly because as Cynthia has said, it has such an important role to play in estrogen metabolism. So if we are really putting strain on that pathway, then we're not going to have enough available for our important PAPs. And we need our SULTs to work. So all these sulfotransferases, SULT1A, 1,2, 1A3, that's how PAPs works. So if anyone presents with multiple SULT mutations, you've got to be considering that this whole PAPs is going to be dysregulated. And Sterling spoke to you a bit about in the last session just how important that these SULT mutations are for overall health. Now, decreased sulfur is affecting serotonin because if we've got a reduced supply of sulfate, it's going to impair the sulfation of serotonin, which then will impair its inactivation. So what we will see is more of an increased serotonin. And we often see this, as Sterling has already said, in autism, decreased social bonding, sensory hyperresponsiveness, seizures, and even motor changes. Now, the SULTs 1 and 2 I predominantly had a look at. Certainly in the fetal brain, it's really huge. It's present in so many different tissues all through the body, but the 1A particularly in the fetal brain. And as you can see, it really targets really important pathways. So if you have a look, it's phenols and our catecholamines, that's why we're talking about it today, but also thyroid, hormones, xenobiotics, and our estrogenic steroids. And if we've got a decreased 1A, it might be protective by forcing some of these toxic metabolites to be processed via another pathway like the glucuronylation rather than sulfation. It'll spare sulfate, but the problem is we're going to have a worse effect to phenols because we're not going to be able to break them down. That will then also affect our phenolic amines like dopamine, tyramine, and serotonin. And it's this SULT1A that's probably the most relevant in autism because it does create this increase in neurotransmitters and the catechols. And often in autism, we'll see increased urinary sulfates but decreased plasma sulfate. The SULT2, they sulfonate cholesterol and hydroxysteroids like pregnenolone, DHEA, and neurosteroids. So that's another key effect that having a reduce in these SULTs will affect because we're not going to have enough PAPs available. Also, another really important use of sulfate in the body is we supply sulfate to our glycosaminoglycans, our GAGs, which we need for cell signaling, fibroblast growth factor, axon growth, brain development, gut function. So even that will have a huge impact on our digestive system. If these sulfur glycosaminoglycans are reduced, then we're obviously going to have inflammation, as Cynthia has already said, and increased gut permeability. Another interesting, vitamin D regulates our sodium sulfate transporter or co-transporter. Increased androgens, it's due to a disturbance of sulfotransferase activity. I cannot say that today, our SALT activity. And because DHEA, remember, is stored as DHEA sulfate. That's what we're actually checking when we're doing bloods. We're checking DHEAS, which is DHEA sulfate, which is inactive. So if we've got a reduction in sulfation, we've got less DHEA converted to DHEA sulfate. And so more will be converted to testosterone. And we already know that testosterone is going to have an effect on CBS, and that will cause reduction in glutathione, increase oxidative stress. So it's all very, very intertwined. And I think certainly, as Cynthia's just mentioned, our eNOS, its main product is sulfate. And sulfate combined with cholesterol will produce cholesterol sulfate in the skin. That's transported to all tissues. And so you're going to have a neurological inflammation if you have got this reduction in PAPs or the reduction in the availability of your sulfate to then be used by PAPs. Now, if we have a look at some of our MAO inhibitors, and I guess the important thing to look is you'll often see someone is homozygous to all the MAOs. You very rarely just see one. You'll see all of them. And as you know, most of our first category antidepressants are MAO inhibitors, MAO-A inhibitors specifically, and they will have an effect on the reduction of the crossing of the serotonin and so increase its availability. However, this really explains why we've got so many people that react negatively to the SSRIs, because it has very much to do with their MAO genes. And when you look at the body's got a great feedback system. When you look at the MAO genes and someone is homozygous for all of them, you think, oh, okay, well, serotonin's going to be too high. But in actual fact, what you've seen over a period of time, that may well have been the case very early on, that you have high serotonin, but the body has a great feedback loop. And what it does, it'll say, okay, I sense that serotonin is really high, and I'm going to down-regulate serotonin production. So by the time you see them, they may well have very low serotonin. But with these people, you've got to be very careful with the dose. And so sometimes a slow-release, low-dose serotonin will just help and I don't know if you all know, but certainly inositol will help the resensitizing of the serotonin receptors, because when someone's been taking an SSRI for a long amount of time, their serotonin receptors are really blunted. So we can help, but we've got to be very careful when we see homozygous male, because initially, yes, their serotonin is going to be high, but they're probably presenting with low. But if you go too hard, too fast, you could send them over the edge again. So you've got to monitor levels very, very closely, and low is always better. So if we have a look, predominantly what we're saying is that if you've got a high MAO activity, so fast MAO, so you're more likely to see that as the green negative, they're going to have low serotonin because they're burning through it much faster. The enzyme is upregulated, and so their downstream metabolites are predominantly going to be, the pressure is going to be on them, but essentially they have low serotonin. There's a few symptoms that we can see, and the problem is that low and high serotonin can often present with some of the same symptoms, but predominantly with low serotonin, you're going to see these symptoms, particularly that obsessive compulsive patient. And I don't mean obsessive compulsive in the true sense of the word, but I mean the people who get fixated on something. They are completely anal in everything they do. They can't move on to step B without first finishing step A. They might get fixated in routines. And you can see it, and particularly those that get worse during the winter months. And carbohydrate craving is a big one. And I'll use this in clinic to say, what sort of foods are you erring towards? Do you have a real craving for carbohydrates? And I really do believe that the body is very intuitive. And if you've got someone that is really craving carbohydrates, don't just think that they just want that lovely feeling, but they really are probably trying to increase serotonin. It's the same as people who love drugs and alcohol and feel great when they drink. You know that they're desperately trying to raise dopamine levels. And so we can get really good clues as to whether their serotonin is high or low, and dopamine, by just asking them very simple questions about their behavior and the food that they're trying to eat. And the more protein, we'll talk about this next week, but part of the problem with these, high protein diets is they're going to increase dopamine levels, and as a result, you're going to get low serotonin. So you've got to be also careful. All these fad diets, they're pushing everyone into this dopamine high, and it's not necessarily the right thing for everybody. And high serotonin, so the low MAO activity, and these are the people that you'll predominantly see as the red plus, or just plus because it's only from the mother. And you can see more aggressive behavior, more antisocial, bit of confusion, agitation, muscle twitching, and often the biggest sign will be GI distress, because they will feel very upset in the stomach. But a lot of the time, this is really hard to differentiate between elevated dopamine. So, probably the biggest key there is that antisocial behavior. They just tend to withdraw quite a bit. So, all right, we've gone through a fair bit today. We'll open up to questions, and I'm sure we've got a few. Suzanne?
We haven't got a lot of questions, I'll be honest. So this is your opportunity to type in any questions that you might have based on the content that's been covered today. The one question that we do have at the moment is from Tara. Now, Tara's actually asking, do the MAO SNPs cause faster enzyme activity or slower?
Oh, well, as we just said, if they've got the red SNP, so if they're the positive, then initially it's definitely going to be an issue breaking down the serotonin. If they're green, they're going to be churning through it a lot faster, and therefore put more pressure on the downstream metabolites and probably be lower in serotonin.
Okay, excellent. Now,
Cynthia and Sterling might have something to add to that.
Okay.
client of mine who, can you hear me?
Yep.
Yep, Cynthia, we can hear you.
So I have a client who is very on top of this, and he's actually seeing another person as well in New York. So he flies around and works on his health, and he got into some problems because he's so focused on getting healthy and doing all the right things, and he's doing a great job. But what happened, and unbeknownst to me, he started to raise his intake of lithium and B12. And he contacted me after he had this whole battery of GI tests because he was having a lot of GI pain and cramping, and he just felt horrible. And so I talked to him for about 45 minutes, and I came to find out that he was really pushing his methylation. He was pushing it way too hard, and I believe that he was driving his serotonin up. And it was causing, because of the vasovagal response with serotonin, he was causing a lot of cramping in his upper GI, and to the point where he almost couldn't eat. So that's something to keep in mind when you've exhausted everything and you've tried everything, and a person doesn't have H. pylori and they've been working on their gut and they don't have parasites and all the things that you would associate, or poor gallbladder function or poor enzyme function. Look at the serotonin.
That's excellent. Sterling, have you got anything to add to that?
Oh, Sterling's muted.
She's muted her end. There she goes.
Not to that one, but I would like to add to the salt intake off where Cynthia was talking about the salt gene and tamoxifen. The salt gene and tamoxifen, I had ran across a few studies the past couple of weeks, and I failed to mention it, is that when somebody's taking the drug tamoxifen, they're fighting breast cancer. A lot of people that are going through cancer, they go through depression, so they're given SSRIs. And people with the SALT1A genes and a combination of the CYP1A genes compromised, when they mix tamoxifen with SSRIs, they have a higher rate of death. So it's not a good mix for them if they're having to take the tamoxifen for cancer treatment to be taking an SSRI with it if they have SALT1As and CYP1As together. I think that's really important for people to know.
Okay, thank you. Sterling, while you're on, we've got another question for you. You mentioned pumpkin seed butter. Are you concerned about the oxalates in pumpkin seeds?
Yeah, if I'm dealing with oxalate issues, but right now I keep mine under control by regularly getting my lysine and B6 myself, and I do a random urine creatinine oxalic acid test every 6 months. And if that was off, yes, I would have a problem, and I would not do the pumpkin seed butter. But as long as I'm keeping that under control, the pumpkin seed butter, it's loaded down with tryptophan. It's very good, especially in the winter months. There's not an oxalate issue.
Okay, we've got another question here from Becky, and I am going to have trouble saying it, so Carolyn, you might want to jump in. It says, can you test a peroxynitrate level?
Peroxynitrate.
Peroxynitrate.
You definitely can't hear in Australia. Cynthia, I'm not sure. Cynthia? Oh, here she comes.
Bowel testing.
Sorry, we didn't hear you because you were muted. Can you just start that again?
There's not a direct marker that I use to look at peroxynitrates, but I look at the indirect markers. So I'm looking for low arginine. I'm looking for high ammonia. I'm looking for the things that some of the analytes that have to do with the CBS pathway, transsulfuration pathway, and then what's happening at the other end with nitric oxide synthase and citrulline, things like that, the urea cycle.
And your organic acid test predominantly tests those key markers.
Yeah. You can see
Can I add something with the...
Yep, go ahead.
Can I add something?
Sure.
We're getting feedback from Cynthia's mic again, sorry.
All right, I'm going to mute Cynthia. Try that.
Yeah, now that sounds better. Okay, again, with PAPs and nitric oxide synthase, if you go read a lot of Susan Costin-Owen's work, she's been studying oxalates for years. They are directly linked, elevated oxalates are directly linked to poor nitric oxide synthase function. So if you feel that there is a nitric oxide synthase problem, again, if you remember what I told you all last week about PAPs and how oxalates shut them down and running their B6 activity, not being able to make ATP. She has a lot of information. Susan Costin-Owen, just all you have to do is do a Google search, put nitric oxide synthase, and Susan Costin-Owen, that's Costenowen's. Just do a search on her, and you'll see a lot of her research on how it directly shuts down nitric oxide synthase.
Suzanne, have we got any other questions?
We have indeed, yeah. We've got one from Becky here that says, can we test quinolonic acid?
Yes, in your organic acids test, they will check markers for both quinolinic acid and kynurenine, and the Great Plains will do that, and so will Genova. And I think, Cynthia,
might I add for people in the United States who cannot afford Great Plains and Genova, there's a lab that almost anybody with health insurance here in America can get 80 to 100% covered through LabCorp, and it's called a random urine oxalic acid creatinine ratio. And if you go and do that lab, you don't want to look at the in-range markers by what the lab goes by, LabCorp goes by. If you see the creatinine on the lower end, but it's not in the red low, and you see the oxalic acid on the higher end, but it's not in the red high, there's still an oxalate problem.
Okay, I wonder, Cynthia, I'm going to unmute you. Have you got anything to add to that?
Yeah, I wanted to mention before we sign off that we talked about the IDO enzyme and the quinolinic acids that actually stimulate these NMDA receptors, much like glutamate. There's a website, and Sterling told me about this. It's called unblindmymind.org for a list of foods that will increase that neuroexcitatory stimulation, and we really don't want to we don't want to overdo it. So unblindmymind.org is good for that. Also, with respect to the SNPs of MAOA and then the receptors, even like for dopamine, MAOA, MAOB, and some of these receptor genes and transporter genes, oftentimes because people have SNPs and because we meet them somewhere along their path of life, you really have to go with symptomology and some test results to determine where they're at, because with neurotransmitters, like Carolyn said, there's these feedback loops, and you can downregulate production and you can downregulate receptors, much like insulin receptors for people who are eating a lot of sugar and, you know, cranking up their insulin until the receptors are just exhausted and say, leave me alone, and then they have to go to like an injectable form of insulin. So receptors and transmitters and carrier proteins work together, and over time they can shift despite the SNPs.
Yeah, good, very good point, because I think it's important for all of these to remember that you can have an upregulation or a downregulation of any single pathway irrespective of your genes, you know, because as we know, when we're talking about epigenetics, it's all about what products speed up or slow down a pathway. So if someone has anything disturbed or they're shunting down too many pathways, they don't even have to have any SNPs to have an issue.
Okay, so the next question then is from Donna. On slide 39, it says something about cancer and BQ. What is BQ?
I'll get Cynthia to answer that because that's Cynthia's slide.
Wait, let me look. Let me look at the slide. Let's see if I can find it. $39.
39, yeah.
39. I'm not sure. Let me see if I can find it. I didn't prepare this slide.
All right, why don't we ask another question while we're waiting?
Okay, well, Donna's got another question. She's saying on slide 41, you mentioned light drinkers, ADN, ALDH, and then heavy drinkers under the CYP2E1. Can you say more about this or explain a bit more about why you said this, please?
Can I go back to the BQ? I'm thinking that means barbecue.
Okay.
Like charbroiled, like maybe I don't know. I don't know. Since we're talking about Q though, I wanted to mention that there's a supplement called PQQ that will downregulate that will help with those NMDA receptor excitations, will help lower them. So it's called PQQ, and it's actually a cofactor in one of the slides. So go ahead, sorry.
Okay, so did you catch the question about slide 41 there, Cynthia?
And we have another one? Okay, let's see.
Yeah. So slide 41, you mentioned light drinkers, ADN, ALDH, and then heavy drinkers and the CYP2E1. Can you say more about this or explain a bit more about why you said this, please?
That's just what happens. People who drink a lot of alcohol will activate the ADH in step 1. It's a step called a gene called ADH. And that requires NAD as a cofactor. Another option is an enzyme called catalase, but for the really heavy drinkers, CYP2E1 is active. And in that case, it requires NADPH, and that's what's happening in the liver microsomes. The CYP2E1 is active. In the catalase case, that's the peroxisomes, which are part of the cells. And then in the ADH, that's your cytosol. But for the heavy liver detoxification of alcohol, it's CYP2E1 to form acetaldehyde, which at that point now downregulates methylation or inhibits methylation. But now you're stimulating your dopamine receptors. And then after that, the ALDHs come into play, and you end up generating acetate and then fatty acids.
Okay, I've got a question here from Janie. She's saying it's an unrelated question, so I'm not sure if you want to answer that during this session. But the question is: are there certain genetic SNPs that should not use curcumin?
Sterling, do you want to answer that one first?
Yeah. People with COX-2 inhibitor issues, mast cell, you should really watch out for. It's a COX-2 inhibitor, and there's many people out there that they've never told you they're allergic to aspirin NSAID, non-steroidal anti-inflammatory drugs, as well as aspirin. Then that would be, or if they've had a history of anaphylactic shock, until you know what's going on with their COX-2. And I hope to have those COX-2 genes out in the near future. You might want to be careful with the curcumin. And if you give it to them very low dose to see their reaction, because not every person, it reduces inflammation. I'm a person that if you give me enough curcumin, I'm going to have an EpiPen, an ambulance, and resuscitation. So it's not a good fit for everybody with COX-2 issues.
No, and it's also a methyl donor too, so you've got to be careful at the doses that you give people. So some people will do exceptionally well on it and others they won't.
Okay. You mentioned, Sterling, you made reference to someone called Susan earlier. And Becky didn't catch the name. Susan who?
Susan Costin Owens. I'll type it in here so you can pass it on to them. Hold on.
Please, yeah. If you type it into the chat box and then select entire audience.
Entire audience. Okay, I'll do that right now.
And then that means everyone will get that. Thanks.
All right. Well, all I see is organizers, panelists, presenter, Cynthia, and I don't see where I have a thing to send it to anybody. But
all right, send it to the organizer, and then I'll relate that out. I'll type that back in here.
Yeah, unfortunately, I don't have that option.
Got it. Okay, I'm just going to type that into the box for you. So it's Susan Costin.
Yeah, she gives a lot of information on nitric oxide synthase and oxalates.
Okay, so that's actually gone through to everybody, Susan Costin Owens. And then in the meantime, we've got another question that's come through. And this is for Cynthia from Scott. Is the PQQ mentioned actually, oh my gosh, Carolyn, you're going to have to say this.
Okay.
Can you see the questions?
Yep, I can.
Quinone, sometimes mixed with CoQ10 for support cellular energy and mitochondrial function.
Yes, I'll let.
Just unmute you, Cynthia. Okay, you're unmuted.
Yeah, PQQ, you can either buy it alone, usually it's 20 milligrams, or you can buy it with CoQ10, where it's 10 milligrams. But like any of these, some of these more unusual supplements, you always have to start low.
Okay, super. That's it then, is it? That was a nice short answer. Right, so just a quick comment here. Donna is saying this is so wonderful. Thank you. Many clinical pearls. And then, right, okay. Donna's asking, can you order the compendium now on the MTHFR website?
We sent out, Donna, a link to it last week with the slides. I will make sure that you re-get that link today.
Okay, super. Well, there aren't any more questions. So is there anything else that any of you ladies would like to say to finish off?
No, I
just wanted to mention, oh, sorry.
No, go on.
I just wanted to mention about the inositol powder. It's very effective, but again, you start low, and the studies that were done with inositol powder use it to elevate serotonin without some of these side effects was 18 grams a day. And the research was in the American Journal of Psychiatry and then another one in the Journal of Clinical Psychopharmacology. And the equivalent of 18 grams of inositol would be like eating 13 cantaloupes and 45 grapefruits, or these huge amounts of fruit. So with the inositol, get the powder and then mix it with something and then work up, especially for people trying to come off of SSRIs. But to date, I've never recommended 18 grams a day, but that's what the studies were done with.
Yes, and I think those studies were also done with people with serious OCD.
Correct.
A lot of the time. So I think 18 grams probably I've used 18 grams in clinic with serious OCD issues. But I agree, just generally, if you want to resensitize your serotonin receptors, it's much better to go lower.
Mm-hmm.
Okay, well, ladies, there's no more questions coming through for you. Like I say, we've got some comments coming through that's saying this is a wonderful, wonderful webinar, so thank you very much. Our next webinar is on Tuesday, November the 3rd at 7:30 for US. And for Sydney, please make a note of this. Because the times change in the US, we will start 1 hour early. So our date will be Wednesday, November the 4th at 10:30 in the morning. So that's 10:30 in the morning. And then all subsequent webinars after that will be will be starting at 10:30 a.m. So our next webinar, webinar 7 in the series, is neurotransmitter, glutamate and GABA. So that's what you'll be able to look forward to for next week. And I'd just like to say thank you once again to everybody that's attended, and again to Sterling, Cynthia and Carolyn for providing such amazing research and information to us. So thank you very much everyone, and good night.